Hello, I'm Claire Burgess. I'm in Daryl Cotton's lab at the Center for Regenerative Medicine at Boston University. And today I'm going to be talking about my project, which is trying to make human alveolar epithelial type 1 cells using iPSCs. So the first question, of course, is what exactly is an alveolar epithelial type 1 cell? So the distal part of the lung called the alveolus is where gas exchange takes place. And there are two alveolar epithelial cell types. They were creatively called the type 1 and the type 2 cell. So the type 2 cell is cuboidal, it produces surfactant, while the type 1 cell is this incredibly thin, flat cell that allows oxygen to diffuse through it into the capillaries. So what we know about where type 1 cells come from mouse lineage tracing studies. So we do know that type 1 cells can be derived from mature type 2 cells after injury and during homeostasis, and that during embryonic development, they are derived from an NKX2-1 positive lung epithelial progenitor cell. So the Cotton Lab has developed this differentiation where we can take iPSCs through several known developmental stages into these NKX2-1 positive lung epithelial progenitors, which we can then sort out and replate in 3D that grows these 3D monolayered epithelial spheres, turn on SPC, and we call those our IAT2s or alveolar spheres. However, while these cells this is bulk RNA sequencing, you can see our IAT2s do express high levels of type 2 cell markers similar to these sorted adult type 2 cells, but they don't express many type 1 cell markers. So they seem to be just made up of the type 2 cells. So I am trying to use this differentiation to make iPSC-derived type 1 cells, but also to understand what are the signaling mechanisms driving this type 1 cell differentiation from the lung progenitors or the SPC-positive IAT2s. So the main pathway that I have been studying is the hipposignaling pathway, specifically its nuclear effector protein YAP. And so the main thing to know about YAP is that when it is phosphorylated, it's sequestered in the cytosol and it's degraded. Whereas if it is not phosphorylated, it is able to move into the nucleus where it will bind to transcription factors and regulate gene expression. So in order to study the effects of YAP specifically on our IAT2s, I cloned these lentiviruses. So the main one I'll be talking about is this YAP5SA. So it's a mutated form of YAP that cannot be phosphorylated. So it will always go into the nucleus and always affect transcription. And it has this tag BFP reporter as well as a LOXP site, so that I'll actually be able to pre-excise it once it is integrated into the genome. And then the control virus that I have is interesting. It's this wild type YAP. So what I see with the wild type YAP that can be phosphorylated is when I infect our IAT2s, that is actually sequestered in the cytosol, I see no effect on gene transcription, and it's very similar to cells that have no virus or cells that have been infected with just a GFP control virus. So just to show you what this does to our cells, I used a cell line called SPC2B2. Kostas Alexandrotis works with that, and it has an SPC TD tomato reporter in it. And I infected these cells with either the YAP5SA or the wild type YAP, and then plated them in 3D matrigel and kept them in our normal sort of distalization media. And so this is what the cells look like. Even 3 days post-transfection, you can start to see differences in these cells that become even more prominent by day 7, whereas our control cells grow as these beautiful monolayered epithelial spheres that have very clear lumens. The YAP5SA is growing in much more aggregated clusters, and even by day 7, you can start to see that it's maybe starting to lose some of that SPC TD tomato expression that's even clearer by day 13 post-transfection. So then to look at what this is doing transcriptionally, I looked at whole well qPCR over this same kind of time series. And so the first thing I looked at were downstream YAP targets. So incrin D1 and CTGF are just very well known targets of nuclear YAP. And what you can see is that CTGF at least is significantly upregulated at day 3, and that one seems to be relatively stable in the YAP5SA population. Whereas incrin D1, which is also significantly upregulated already at day 3, continues to climb over this 13-day time period. Our type 2 cell marker SPC, it's already lower at day 3 and continues to drop off towards 0 as the time course goes on. And then lastly, I'm going to look at 3 different type 1 cell markers. So here we have CAV1, AGER, and potoplanin. You can see that potoplanin may be lagging a little bit behind AGER and CAV1. It's not really different until 5 days post-transfection. But particularly with CAV1 and AGER, you can see that they're still climbing even at this day 13 time point. They have not quite reached a plateau of yet. So they may, if I took it beyond this day 13 time point, they may go even higher. But like I said, this is sort of bulk qPCR, and presumably not all of the cells in each of these wells was transfected with the lentivirus. So to understand a little bit more about the cell heterogeneity, I performed single cell RNA sequencing. I chose the day 7 time point because we know the most about that from our IAT2 control population. And you can see that all of the type 1 cell markers that I've studied are already turned on by that day 7. So to show you kind of what this is going on with the SPCTD tomato, this is what the cells look like when I took them to the sorter. You can see the wild type YAP population is maintaining that SPCTD tomato expression, while the YAP5SA seems to have kind of split into 2 populations, one that is SPCTD tomato positive and one that is negative. So I did not sort on this SPC. I just sorted on live and then sent them for the Tenet single cell RNA sequencing. And so this is what that looks like. It's a spring plot, which is a force-directed 2D projection of the single cell RNA sequencing data. A huge thank you to Carlos for putting this together for me. And what you can see is that the wild type YAP control well seems to have all clustered together, regardless of whether they got the lentivirus, while the YAP5SA population has clustered into 2 separate populations, one that is with the wild type YAP, again, presumably IAT2s, while another that is clustering completely separately. So presumably this is due to the effects of the virus on that population of the cells. And just to show you why you should believe that is looking at these YAP downstream targets, particularly Anchor D1 and CYR61 do seem to be exclusively turned on that in that YAP5SA cluster. And it seems to be that the YAP5SA is driving this separate cluster in a sort of cell autonomous manner. The cells that got the YAP5SA are the cells that are clustering separately. And then just to show that that wild type YAP cluster is our type 2 population, they express SPC NAPSA, as well as this AT2 gene signature that was developed using a human primary single cell data set by Haberman and Kropsky that came out in 2020. And you can see that this wild type YAP cluster is very high in that type 2 cell gene signature. And then when you look at our type 1 cell markers, AGER, CAV1, CLIC5 are all highly enriched in that YAP5SA driven cluster. And potoplanin, again, may be lagging a little bit behind, but it is still exclusive to that YAP5SA cluster. And then looking again at that primary derived type 1 cell signature, you can see that that has been turned on in exclusively this YAP5SA cluster, while the type 2 gene signature has been turned off. So to look at this in a little bit less biased way, using Louvain clustering, this is a heat map of the top 50 differentially upregulated genes in each of the 3 clusters here. And you can see this light blue cluster 0 is at the top here. Our top genes being type 2 cell genes, SPB, SPC, NAPSA, even the TD tomato is on there, while this yellow cluster 2 down at the bottom here is mostly driven by genes like TOP2A and KI67, so proliferation associated genes. So I've called those clusters the IAT2s and the proliferating IAT2s. And then this cluster 1, that's the YAP5SA cluster, the top differentially upregulated genes there are YAP downstream targets like Anchor D1 and CYR61, as well as type 1 cell associated genes, CAV1, CLIC5, AGER, EMP2, and several others. So it does seem to be that the YAP5SA is working in a cell autonomous manner to drive this type 1 cell program. And I wanted to compare these cells to primary cells. So I put them together with, again, that sort of Haberman-Kropsky single cell data set. And so what this graph is showing you is the Kropsky epithelial cell population, so all of the lung epithelial cells, and then my iPSC derived single cell data. And the way you read this is the larger the circle, the more cells in that population that are expressing any level of that particular gene. And then the darkness of the purple is associated with how what the actual expression level is. So the deeper the purple, the higher the gene expression is. And what you can see is my IAT1s compared to this primary AT1. The size of the purple dots, or of the purple dots, is relatively similar. Again, potoplanin may be lagging a little bit behind. You can see that the coloring is lighter in my cells, but again, this was day 7, and I showed you the time series showing that the AT1 marker expression seems to only go up over time. So presumably, if I were to redo this at a much later time point, that I might get darker purple dots on that. Another thing to note is looking at these YAP targets, they're very dark, very large in my IAT1s. However, they don't show that same expression pattern in the primary type 1 cells. And then again, looking at proliferation markers, there is a small subset of my cells, the IAT1s, that do seem to be proliferative, which is not something that you would see in primary type 1 cells. But it does seem to be that this YAP5SA is turning on this type 1 cell program, and it is the type 1 cell program, not a transitional cell or one of these like aberrant transitional cells. It seems to be specific to the type 1 cell program. But in order to study this further and maybe get away from a lentivirally based differentiation protocol, I wanted to be able to track and purify these cells as they're turning on this type 1 cell program. And so to that end, I have generated a reporter iPSC line. So this line carries an NKX GFP for tracking lung epithelial progenitors. And then I used CRISPR-Cas9 to target the TD tomato to the start site of AGER to be able to track these IAT1-like cells. And so just to kind of show you proof of principle, I differentiated out this cell line to our NKX positive sort of distal cells, and then infected them with either the YAP5SA or the wild type YAP lentivirus, plated them again in just our 3D matrigel in our type 2-like media. And what I saw was after 3 or 4 days in culture, I was actually able to see this TD tomato turning on in these cells that again still have that kind of aggregated phenotype. And then more importantly, I was able to see it on the sorter and actually be able to sort out these AGER TD tomato positive cells. And so to show you that the tomato is enriching for these type 1 cell markers, on the next slide, I'm going to show you some qPCRs, but keep in mind the wild type YAP is completely unsorted. YAP5SA is this completely unsorted graph. And then the TD tomato positive is this kind of double positive cluster, and the tomato negative is this single GFP positive cluster here. And so if you look at the type 2 cell markers, we can see that the tomato positive sorted population is depleted in both SPC as well as ETB5, which is a transcription factor associated with type 2 cells. And then looking at type 1 cell markers, we can see that it is enriched for AGER, potoplanin, CAV1, CLIC5, and HOPX, while both tomato positive and tomato negative are both enriched for NKX2-1, as they were both from that GFP positive population. So this is hopefully showing that AGER TD tomato reporter is working, and that we can maybe use this to isolate out these IAT1 cells and study them further, as well as look for a way to turn on this cell program using something other than a lentivirus. So the future directions or things that I am planning to do is try to really understand the role of nuclear YAP and hippo signaling in this type 1 cell differentiation, really trying to understand what are the signals that's driving this nuclear localization of YAP, because it does seem to be, you can overexpress as much wild type YAP as you want, but if it's not going into the nucleus, it's not going to cause this differentiation that I'm seeing with the YAP5SA. So whether that's cell signaling or mechanical stretch that's really driving that nuclear YAP, I'm going to figure that out, as well as understanding what YAP is really interacting with in the nucleus to drive this type 1 cell fate. And then lastly is optimizing this type 1 cell differentiation using my NKX GFP AGER TD tomato reporter line, hopefully finding a more media-based IAT1 differentiation protocol. So of course, a huge thank you to Daryl and the rest of the Cotton Lab, and the rest of the CRIM and BU and the Pulmonary Center, and particularly Brian Tilton for sorting and Carlos for his bioinformatics. Thank you.